Hello class, this is a recap of the lab that we did, where we looked at the plant cells. So the wonderful thing about an onion is that you can get a layer that is precisely one cell thick. And this is essentially what you saw here. What we're able to really see clearly is a really clearly defined cell. Now, as you can see, they're approximately rectangular shape. They all vary, but they have a very consistent kind of shape. So just like every human is diverse, we all have similar structures and features, but we have variation. We see the same thing here. Now, it's difficult, but you can see hints of organelles inside. What we did was we used iodine. By placing iodine onto the cell, the iodine was absorbed into the cell. The iodine was actually able to go through the cell wall and through the cell membrane, and we were able to see that certain organelles absorb the iodine in greater amounts. So what we were able to see really clearly was still see the cell wall. And if you're really, really carefully observant, you were able to see inside something that would resemble the cell membrane outside of the cell wall. And then inside you could see one, maybe two dots. I think that the two dots probably mean that there's a different organelle that is being shown, but you can see the largest, thickest dot is going to be the nucleus. The nucleus is able to absorb the iodine to a greater degree than any other organelle. So your lab should be showing, when you draw, you should be attempting to accurately capture individual shapes. See how each individual shape is different. I do not want to see something like that, or that is not an accurate representation. That is a symbol and not something that I want to see. We want to see individual, unique shapes that attempt to capture the reality that you see in front of you.